Today's topic is about biochemistry of AIDS and HIV. In detail of this, we will look. So first about the epidemics of AIDS. In 1981, a cluster of 5 cases of pneumocystic carinine pneumonia were reported in USA. These protozoa can produce pneumonia only in immunodeficient individual. It is not generally producing infection in a normal person. Based on that clinical manifestation, the disease was named as a acquired immunodeficiency syndrome. Means a immunodeficiency is developed in that person. So the virus was originally designated as a HTLV3, but later on is a redesignated as a HIV, means human immunodeficiency virus, in 1986. So what about Indian scenario? The virus found to be entered in India in 1980. From 1986 onwards, the ICMR has started the serosurveillance against this virus. So, the first seropositive individual in India were identified in 1986. The government of India has established the NACO, that is National AIDS Control Organization, in July 1992 for the prevention of these AIDS. The adult HIV prevalence in India is now in a declining phase. So that is about epidemics or starting phase of the HIV in India. Now, what about the transmission of this virus? 80% is sexually transmitted, 50% transmitted to the blood, drug abuser, frequent blood transfusion, where frequent blood transfusion needed as a part of treatment, mostly in a hemophilia patients. 5% transmitted from mother to fetus through the placenta. But about only 30% may get the infection of HIV. Rest of them may not get infection. So that about the transmission of this virus. What about virus replication? The virus has an extraordinary rate of replication. Over 10 billion particles are produced per day inside the host. So extensive limit of this virus replication there are, but transmission is through the mostly sexually, blood transfusion, or from mother to the fetus. Now what about the natural course of the disease? So it is divided in a different period. First, that is window period. When the virus enters the body, it is multiplied in the body cell but cannot be detected easily. The viral capsid antigen P24 can be detected in the blood during this time of the window period. Now next, that is seropositive stage. After a few months, antibody are seen in the circulation. This is called a seropositivity about this disease. The person is completely normal but transmit the disease to other in the seropositivity condition. Now about this seropositive individual progress to the third stage of the disease. So in that about 10% within 5 years, 50% by 10 years, and 90% by 15 years. But that duration of progression to the third stage, that can be extended if proper treatment is given, and if proper treatment is not given, then 90% may get or progress to the third stage within 15 years. For each AIDS patient, it is said that 100 seropositive person in the general population. So what about AIDS disease? When the clinical manifestations start, that is called AIDS disease. Here immune status is depressed. So due to that, microbes will start multiplication and that person is succumb to death within about 2 years. This particular immunodeficiency leads to the fungal, viral, parasite and bacterial infection. Cancers like Kaposi sarcoma and non-Hodgkin lymphoma is also seen. Cognitive impairment due to the meningitis, neuropathy and neuropsychiatric manifestation may occur in a later course of the disease. So further about clinical presentation, lymphadenopathy or enlarged lymph node and fever is seen. AIDS related syndrome are widely, mainly that immunodeficiency, and that is leading to multiple infection. So in detail, microorganisms enter into the body and produce lesion in various systems because they are not destroyed by the immune system. Gastroenteritis and tuberculosis predominantly found in the India. Weight reduction is also seen along with this AIDS disease. So what about immunoselection of the virus? In the early phase, the HIV enters macrophages of the body through a receptor jointly made by the CD4 and CCR5 protein on the surface of the macrophage. So first targeted cell of this virus, that is macrophage. Over that macrophage, these two receptor molecules present, CD4 and CCR5. The HIV surface antigen GP120, that is glycoprotein 120, has a perfect fit only for this type of receptor on the macrophage. So first targeted cell, that is macrophage, and that is called M-tropic HIV, M for the macrophage tropic HIV. Further about this immunoselection. So in the early period, lymphocytes are not affected by this virus. But later on what happened that we will look. So once there is a macrophage is affected, after a few years, the GP, glycoprotein 120 of this particular virus antigen is mutated so that dual tropic virus are produced. So in the late phase of the disease, T-tropic HIV is generated. Previously there is a M-tropic, macrophage tropic, now later on T-tropic. By this time, all the GP120 molecule are mutated such that they can fit only with the T lymphocyte receptor made up of CD4 and CXCR4 protein. So first it is targeting CD4 and CCR5, later on both, and finally it is targeting particularly T lymphocyte. So it is called T-tropic HIV. So the T helper lymphocyte are preferentially killed, leading to the disease manifestation. So in later stage of the disease, particular T cell are targeted and destroyed in the body. So it is shown in this picture. First, that is M-tropic virus transmission. So it is affecting mostly macrophage by the CD4 and CCR5 receptor. Later on dual tropic virus, and later on it is transferred to the T-tropic virus, where particularly CD4 plus CXCR4 receptor are affected. So it is first affecting macrophage, later on it will affect T cell of the body. So here exactly how it is occurring. So free HIV that is able to attach this GP41 which is present over this HIV virus. So these attach over this CD4 receptor of the cell, particularly macrophage, and later on if CACR5 both is combined, and that is helping to perfect fit of this particular virus over the cell surface. Once it is attached perfectly over the cell surface, then it is internalized inside the cell. So that is about the CD4 and CCR5. Sometimes genetic resistance against AIDS is also found. That is interesting. About 1% of population are resistant to the HIV infection. Here CCR5 and CD4 protein together form the receptor for HIV. But actually what happen in genetic resistance, people having mutated CCR5 gene, the protein is not displayed on the macrophage surface. This particular receptor is not present, so it is giving advantage. So without the CCR5 protein to latch into, HIV fails to invade macrophage. So this HIV virus is now not able to enter inside the cell. Thus an individual with mutated CCR5 gene escape the HIV infection, and that is one kind of advantage. Laboratory diagnosis of AIDS. It is possible with the help of ELISA method. Antibody against only one antigen, particularly glycoprotein 120, and here there are more chances of the false result. In detail also we will discuss later on in this video. Another laboratory diagnosis, that is Western blot. Here antibody against 6 different viral component is used, so it is more confirmatory as compared to ELISA. T helper count, normally it is more than 400 per cubic millimeter, but it is decreased as per the AIDS disease. RT-PCR, number of HIV particle less than 5000 copies per ml of blood has good prognosis, monitored during treatment per course. So depending on this HIV particle, the different modalities available. Nucleic acid testing, amplify and detect specific sequence of HIV gene, mostly that GAG, POL and ENV of gene of this particular HIV virus. Detection limit 200 to 400 copies per ml, and shorten the window period. We can identify as early as possible. So in detail of that, mostly screening test that is used as a ELISA method that can give result about 2 to 3 hours. So false negative result, more chances because of the window period. If person is window period, then there are chances of coming of a false negative result. In this test, antibody against only one antigen, mostly GP120, is being tested. So there is possibility of false result. Now second, that is Immunoblot. So here particular 6 different antigen can be detected with the help of different method, blotting, electrophoresis or antigen detection method. Now next, that is T helper count, and that is by the flow cytometry. Normal level is more than 400 per cubic millimeter. In AIDS patient always below 300, but if disease is progress, the helper cell count is correspondingly lower down. Below 200 is considered a bad prognosis for that person. So provisional DNA, proviral DNA, that can also detected with the help of HIV-DNA PCR, that is polymerase chain reaction. In this stage sensitivity is 95% and specificity, that is 97%. Diagnosis during window period is also possible by detecting proviral DNA. Intermediate Western blot result that is also used differentiate between HIV-1 and HIV-2 infection by this testing, and another method that is RT-PCR, that is real-time PCR, where number of HIV particle in blood that can be detected. So that is quantification of HIV RNA in plasma, free viral load assay, helpful in monitoring progression of disease as well as efficacy of antiretroviral therapy, whether that after giving treatment viral load is maintained or it is in a progression phase, that can be detected by this RT-PCR. A value of less than 5,000 copies per ml of blood has good prognosis, while a count more than 1 lakh per ml means very bad prognosis for that patient. Now, further about detection of P24 antigen, it is useful in equivocal Western blot result to detect infection during window period, while disadvantage is that it is expensive, as well negative test does not rule out the infection. So even in the screening test some doubt is there, then sometimes the detection of P24 antigen is done here, that is poorly quantifiable, because of that P24 may not be detected sometime in the sample. And lastly, one more laboratory test, that is nucleic acid testing. So amplification and detection of one or more specific target sequence located in a specific HIV genes like GAG, POL and ENV with the help of this nucleic acid testing. Detection limit is 200 to 400 copies per ml. By this test, window period is shortened to less than 2 weeks. Even in the window period, we can detect this particular nucleic acid. So critical because during the early state of infection, people with HIV can be 30 times more infectious, because even in the early phase we can detect and we can prevent that disease progression with the help of this testing. Further about this HIV virus, the structure based on that retrovirus group. Two copies of single stranded genomic RNA is present in the HIV virus. Mostly protein component like P32 is present. So detail of this structure that we will look here. A structure of HIV virus is shown. Inside there is a RNA molecule is present. So what about the nomenclature of this different part of this HIV that we will look? It is here that number is given. So first about this one number, that is showing a nucleocapsid. Further, there are two particular enzyme is present surrounding this nucleocapsid. One that is reverse transcriptase and third one more that is integrase. So these two enzyme is present surrounding this nucleocapsid. This nucleocapsid is covering particular viral RNA genome of this HIV virus. So viral RNA genome, nucleocapsid, and surrounding these two enzyme, integrase and reverse transcriptase. Further, there is a particular gene like VIF, VPR, NEF and P7 is present inside this capsule, and this capsule is called as a capsid. And further, beyond this capsid there is a one more layer is that is called matrix. In between this capsid and matrix, one particular protein is present that is called a protease. So this protease enzyme is also important part of this HIV virus. Further, beyond this particular protease, a lipid membrane is present, and in this lipid membrane particular two antigen is present, and that is called a GP41, that is transmembrane glycoprotein. So that is transmembrane, and one more antigen that is GP120, that is called docking glycoprotein. So with the help of mostly this GP120 that is attached over the receptor over the host cell, and that is this virus is able to enter inside the host. So that is whole about the structure of the HIV virus. Now next, that is about virus entry. GP120 of HIV binds with the CD4 molecule on the surface of target cell. CD4 acts as a receptor for the virus present on the surface of the T helper cells, and therefore receive maximum attack of HIV when it is attacked by the T helper cell. Replication of HIV, viral RNA act upon by reverse transcriptase. A complementary of DNA is synthesized. This proviral DNA migrates into the nucleus of the host cell, then integrated with the help of viral integrase. Once it is integrated in the host DNA, then multiple copy of this virus is generated. So retrovirus do not replicate in non-dividing cell, requires activation and division of T cell for the viral particle synthesis. So viral genes are transcribed and translated by host cell mechanism. Later, the GAG and POL genes produce GP120 and GP41 for the encoding of this virus, and that will be released into the surrounding. So these are then enveloped and on the virus DNA. So finally it will be released. So here exactly how it is occurring, that is seen. shown in this picture. So infecting HIV virus. So there is a projection GP41 and GP120. With the help of CD4 receptor, it is attached over the cell surface. Once it is entered inside the cell, this viral single-stranded RNA is released into the cytoplasm. With the help of reverse transcriptase enzyme, which is present in this virus, that is replicating and making a DNA strand. Once it is RNA DNA strand is generated, then it is further converted into the double-stranded DNA. Now this double-stranded DNA, viral DNA, that is incorporated in a cell nucleus. So that is provirus, which is generated inside the nucleus of the host cell. Once it is inserted in a provirus, further from this provirus, mRNA and from that protein is synthesized. So finally this protein, that is making envelope protein like GP120 and GP41, and with the help of this envelope protein, different virus that is released into the surrounding, and finally new HIV particle is released from this host cell, or sometimes this host cell will be bursted out and release lots of amount of this virus into the surrounding. So that is about the life cycle of the virus. Now about HIV genes. 3 structural genes like GAG, POL, ENV, 3 regulatory genes like TET, REV and NEF, as well as 5 accessory genes like VIF, VPR, VPU, VPT and TEV or TNV. In between long terminal repeats are present. So these out of different genes, you have to remember about the GAG, that is for the capsid antigen formation, POL gene for the reverse transcriptase enzyme production, and ENV for the surface antigen, that is G120 formation. These 3 are the most important inside the HIV virus. Now what about immunology of AIDS? HIV preferentially enters into the T helper cell and they are lyse. These cells play a pivotal role in immunological system. Lyse helper cell leads to the T killer cytotoxic effect reduce. So immunity are thus paralyzed and pathogens get entry in the body. So because of that, immunity is affected because that T cell is now not able to work, particular T helper cell as well as T killer cell. So our immune system is down, and due to that lots of pathogens or virus, bacteria or fungus that can enter and multiply inside the body. So that is the main affecting our immune system. Genetic heterogeneity of the virus. 2 type of HIV virus is seen, HIV-1 and 2. HIV-1 is more virulent, infective and predominant. Many subtype of HIV-1 is also found. Different subtype may be present in a one patient. High mutation rate in a virus of this HIV-1. About 50% of amino acid in the envelope GP120 are highly variable. Therefore, the antigenicity of the virus also varies. The virus can escape from the immune attack. So because of that, it is that particular virus is not affected by our immune system, they can multiply enormously in our body. So about this species HIV-1 and 2, the high virulence infectivity is seen as well as prevalence is global for this HIV-1, while HIV-2, that is lower virulence and infectivity, but it is mostly seen in only in West Africa. So that is about the genetic heterogeneity. What about anti-HIV drugs? So one, that is nucleoside analog, and that is working as a reverse transcriptase enzyme inhibitor, which is the main enzyme of this HIV. This particular analog inhibit RT reverse transcriptase of the HIV. The drug used are azithromycin as well as dideoxyinosine. One more RT inhibitor or reverse transcriptase inhibitor, they are the non-nucleoside analog. Previously nucleoside analog, here non-nucleoside analog. This agent make the enzyme inactive, that is reverse transcriptase to make inactive, like nevirapine and delavirdine. Further, protease inhibitor extremely selectively block final assembly and package of HIV particle and release into the circulation. So that is ritonavir and indinavir. So nucleoside analog, that is reverse transcriptase inhibitor, RT inhibitor, non-nucleoside analog and protease inhibitor. Further, some drugs that are also under experiment like RNA interface, that is used based on the gene silencing by using this synthetic small interfering RNAs. So that all in the experimental phase. One more, that is prevent entry of this HIV virus into the cell by CCR5 co-receptor antagonist used to prevent interaction of this GP120 and CCR5. So if we can prevent entry of this virus inside the cell, we can also prevent the infection. So by this antagonist that can be used as a treatment part. So mostly when it is treatment is given, a combination of drug that is called a HAART, that is highly active antiretroviral therapy, reduce viral load and prolong the life of the patient for the many years. So we can prolong that lifespan of that person once it is get infected if it is given a proper drug. So that is about the anti-HIV drugs. But prevention is always better than the cure. So education and awareness, all the blood samples should be tested before blood transfusion for this HIV virus. Surgical instrument properly sterilized to prevent cross infection. Boiling for 10 minutes, that will inactivate the virus. Blood spills by hypochlorite, that can also destroy the virus. Heat sensitive instrument may be decontaminated by immersing in a 2% glutaraldehyde. So these all are working as an antiviral agent and they can destroy the virus. So we can use that all antiviral agent properly and we can prevent infection to the other person. So that's all about the HIV or AIDS. So basic biochemistry of HIV and AIDS is explained in this video.